Welcome back. Today, we're diving into a fascinating genetic detective story. So imagine this. You've just tried to give a new superpower, a brand new gene, to millions and millions of bacteria. The problem? Only a tiny fraction of them actually accepted it. So how on earth do you find those microscopic few, the successes, among a literal sea of failures? That's the case we're cracking today.
Really, think about that for a second. This is the central challenge. You can't exactly line them up and check each one. We're talking about a biological needle in a haystack problem on an epic scale. So where do you even begin?
And this is what that haystack looks like. Just look at it. Every single one of those little dots is a colony, made up of millions of individual bacteria. Somewhere in that massive crowd are the special ones we're looking for. The question is, how do you spot them without losing your mind?
Well, every good detective needs their tools, right? In genetics, the first step is getting your gene of interest into the bacteria. But once you've done that, the real investigation begins. That's the screening process, finding the ones that worked. And that gene delivery happens using something called a plasmid. The easiest way to think about a plasmid is like a tiny biological USB drive. Scientists load their new gene, their software update, if you will, onto this little ring of DNA. And then they offer it up to the bacteria, hoping they'll take the bait and install the new program.
So let's go back a bit. How did scientists first crack this case? Well, they came up with a really clever trick, almost like a two-password security system that would filter out all the bacteria that didn't get the right genetic upgrade.
This is one of those early plasmids, a classic called PBR322. Now, you don't need to memorize all the little details here. Just focus on one key feature. It naturally carries two of these passwords. And what are they? They're genes that make the bacteria resistant to two different antibiotics, ampicillin and tetracycline.
And here is the genius move. Scientists designed this plasmid so that the only place you could insert your new gene was right in the middle of that tetracycline resistance gene. So, if the new gene goes in successfully, guess what happens? You break that password. The bacterium can no longer fight off tetracycline.
Okay, this table lays out the clues perfectly. The bacteria we actually want, the recombinant ones that have our new gene, they'll survive ampicillin, no problem. But because we broke that second password, they'll die if they see tetracycline. Now, any bacteria that just grabbed an empty plasmid, what we call religated, they're going to survive both antibiotics. They're the imposters.
So here's how the actual lab work went down. First, you grow everything on a plate with ampicillin. That kills off any bacteria that didn't pick up a plasmid at all. Then comes the really tricky part, replica plating. You take a stamp, make a perfect copy of your plate, and transfer it to a second plate, but this one has tetracycline. The final step, you hold them up side by side. Any colony you see on plate number 1 that's gone from plate number 2, that's your winner. It's brilliant, but wow, you can imagine how tedious that was.
Yeah, that whole two-plate system was a massive amount of work. So, science did what it always does. It found a better, more elegant way. A method so clever that the bacteria literally change color to tell you if they've got the gene. This is called blue-white screening, and it's a total game changer.
And the cool thing is, this new method uses the exact same core principle as the old one, insertional inactivation. The idea is really simple. If you stick a big chunk of new information right in the middle of a working gene, you're going to break that gene. It just stops working.
And would you look at that beautiful result? No more comparing plates. The case is solved on one single dish. The white colonies, those are our successes. They have the gene we want. The blue ones, they're the failures. It's an instant visual clue. So much easier.
So how does this color-changing trick actually work? Well, it all comes down to a newer, smarter plasmid, like this one called PUC18. Instead of a second antibiotic resistance gene, this one has a special gene called LAC-Z. You can think of LAC-Z as a tiny paint factory. When that factory is up and running, it makes an enzyme that turns a chemical in the dish bright blue.
And you can probably guess where this is going. Just like before, the spot where we insert our new gene is plunked right down in the middle of that LAC-Z gene. So, if our gene gets in there successfully, it breaks the paint factory. The machinery gets shut down, the colony can't make the blue color, and so it stays white. It's just an incredibly clever and efficient solution to that whole needle in a haystack problem.
Okay, so now we can find the right bacteria super easily. That problem is solved. The next logical step for scientists was, how can we make these tools even more powerful and versatile? And this led to a whole series of awesome upgrades for the genetic toolkit.
One of the best upgrades is something called the Multiple Cloning Site, or MCS. If the original plasmids had just one specific spot to insert a gene, like a single picky keyhole, the MCS is like a master keyring. It's a short stretch of DNA that's packed with a bunch of different insertion sites, giving scientists way more flexibility to use whatever genetic key they happen to be working with.
But just having the gene sitting inside the bacteria isn't always enough. You often need to tell the bacteria to actually use it, to turn it on and make the protein. That's the job of what's called an expression vector. It's a plasmid that comes with all the control signals built right in. A promoter, which is basically the green light to start reading the gene, a terminator, which is the stop sign, and other key signals. It's the complete instruction manual.
And what you're looking at here is a supercharged version, a T7 expression vector. You don't need to know all the details, just its purpose. Its control system is so powerful, it's like putting a turbo engine on that gene. The whole point is to turn that one little bacterium into a microscopic factory completely dedicated to churning out massive quantities of whatever protein you've told it to make.
So, in just a few big leaps, we've gone from a complicated detective hunt to a simple color test, and then all the way to building these incredibly powerful microscopic factories. But let's take a step back. What's the point? Why does any of this stuff matter out here in the real world?
Well, here's why it matters. This entire process, from that clunky old two-plate system to the elegant blue-white screen, this is the absolute bedrock of modern biotechnology. It's how we make life-saving medicines. You know, the insulin that millions of people with diabetes rely on every single day? It's made in giant vats of bacteria, tiny factories that were found and built using these exact principles.
So we've basically mastered the art of finding one special cell among millions and turning it into a biological production line. This toolkit has already completely revolutionized medicine, and it really just leaves you with one big question. With tools this powerful, what problem are we going to solve next?